conversion into THC and all the different isomers, but there are other conversions that can be made to form other novel cannabinoids that might be interesting for future therapeutic research. So I wanted you to kind of touch on that too.
Yeah, absolutely. So I'm involved in that research right now. And so we've looked at hydrogenation. There's a couple other groups in the world that are looking at hydrogenated cannabinoids. Most notably, you've probably heard about HHC, which is hydrogenated THC. We are the inventors of HHCA, which is basically the carboxylated version of that. You're looking at a vial of it right here. So this is hydrogenated THCA. And you know what's really interesting, Ben, is that our focus is definitely not on recreational products and trying to get hemp-derived things across the goal line. Ours is strictly academic and as researchers. And what we're finding is that anything that THCA can do, HHCA can potentially do better. And so when we make HHCA, when we hydrogenate that ring, we make a mixture of the 9R and the 9S isomer. So by collaborating with Wes Umstead of Chiral Technologies down in Westchester, PA, we were able to basically do a chiral separation to separate the R from the S. And what we're finding, which is very, very interesting, is that the S isomer is more active than the R isomer, which is just the opposite of what we know about HHC itself in terms of its action at the CB1 receptor. What's known about HHC is that the R9R compound is a better binder to the CB1 receptor than is the 9S compound. So obviously, our compound, which is not HHC, it's HHCA, so we have the acid group there, is not a typical substrate for the CB1 receptor, but we're finding that, again, the S isomer is much, much more active than the R isomer. We don't know where it's hitting, so that makes a lot of interesting science. Now we need to figure out, what kind of target are we hitting? And another thing I presented down at CannMed was the chemical transformation that we've looked at now, where we've taken the double bonds of those molecules and not just added hydrogen. Now we've added hydrogen fluoride. So we've done a hydrofluorination of the double bond, and that puts a fluorine on the molecule. And fluorine is super interesting to medicinal chemists because it's small like hydrogen. So it has a very small atomic radii, right? It's up here in the
Hmm.
northeast corner of the periodic table, kind of like where Maine would be. So it's got a small atomic radii, but it's a very powerful electron withdrawing atom because it has no shielding from the outer shells of electrons. So because it's such a strong electron withdrawer, its withdrawal of electron density through the sigma framework really contorts the ring. So I think when we look at the hydrogenated compounds and compare them to the hydrofluorinated compounds, we see a definite twist in the geometry that we think can be manipulated in such a way that we could boost selectivity or boost safety. Or we're still in the beginning of looking at the biological activity of these compounds. But yeah, those are two chemical transformations that we're working on to basically make modified cannabinoid derivatives that we'd like to develop as leads for pharmaceutical companies.
Excellent. Excellent. Well, that's exciting.